So here we go, what we all came for. This morning's keynote focuses on one of the most important shifts happening across AI drug discovery today, moving from isolated AI development toward collaborative intelligence at industry scale. For years, one of the biggest challenges to our work has been getting enough of the right data, high quality data, sufficient variety, and organizations collectively possess enormous amounts of structural biology and molecular data, but there are some competitive concerns to sharing that. So federated learning is beginning to change that equation. Today, we're going to hear from leaders across academia, biopharma, AI infrastructure, computational drug discovery, as they discuss the AISB Network Initiative and how collaborative AI frameworks are reshaping the future of structural biology, molecular prediction, and drug discovery itself. So join me in welcoming the panel. We have Mohammed from Columbia University, Jonathan at Eli Liully, JT from Apheris, Woody at PsiThera, Christina at Bayer, and Armand with SandboxAQ.
All right, great. Well, it's wonderful to be here. Thank you all for coming. So we're going to do things a little bit differently than a typical panel where we're just all up here talking for the whole time. So we're going to give a little bit of background. Each person is going to have an opportunity to talk about their part of the equation, because there's many pieces that have to fit together here in order for this federated learning concept to play from your questions to answers that you need to make decisions in your programs. So let me just jump right into it. So foundation models is where it all starts. We all know about this concept of foundation models, and there's foundation models for lots of things. Language models is what folks are using mostly all day, every day. But there's all kinds of different foundation models for AI, and we want to do a little bit of distinguishing between the different types of models, and here we're going to be focusing a lot on structural biology, and as was mentioned here, there's an AI structural biology initiative where this federated learning thing is coming together, but there's different kinds of federated learning built on different kinds of foundation models, and then once you have a good model, you ultimately want to get into making that model perform as well as possible, and the ultimate goal is, of course, making decisions in drug discovery projects that are going to, or whatever your projects are in the molecular world, to advance your programs. So we have speakers for each one of these areas, and there's going to be some mixing between them as we flow through this process. Each person is going to have 5 or 10 minutes to talk about the specific focus of their research and this focus of their company. And then we're all going to come together up here, and we have a series of questions that we will take you all through, and then there'll be an opportunity for folks from the audience to ask questions as well through Slido. The hope here is by the end, we all have a much better understanding of what this whole process looks like, and moving from a bunch of words and lingo to more tangible components and how this all fits together. Again, our focus is going to be very much on one aspect of this in general, but and that's the structural biology part. But Jonathan will be talking also about ADME modeling, and we can really think about this broader field of foundation models for molecular interactions, for molecular AI, as something that's somewhat distinct from large language models. So for each one of these, we have Mohammed, for example, talking about the foundation models itself. We have JT and Jonathan are going to be talking about this idea of federated learning and how we can actually learn from a whole bunch of proprietary data without actually sharing that proprietary data. And that's really the guts of federated learning. And then we're going to talk about what you do once your models are as good as possible with broad data. How do you start digging into your project specific work, and how do you use those data as your project evolves and accumulates more data? Ultimately, the goal again is to make impact on decisions. It's nice to have all these technologies that are complicated. There's a lot of sophisticated science and components that have to come together to make this work, a lot of scale. But at the end of the day, as was actually just presented before, it's about asking questions that matter and being able to get access to the tools necessary to get answers. So I want to give a little bit of background with OpenFold. So my day job is the founder and chief innovation officer of PsiThera, but I spend mostly my nights and weekends as the chair of the OpenFold executive committee. OpenFold is wonderful. If you haven't heard about it, if you haven't, I suggest looking more at it. But a lot of the folks that are on the panel here actually are either directly or indirectly involved with OpenFold. So just a little bit of background. The idea is to deliver open source tools for the community around structural biology and specifically AI tools more generally for solving biology and drug design problems. We're strong believers in open source, and I think that many fields have evolved in a way where open platforms are essential, and we've seen this across broad spaces, but it hasn't really happened so much in the bio drug discovery space. It's happened with various tools, but we're starting to see it all happen more broadly. And as we start to build these larger infrastructures, we're going to need these open platforms that we can all build on. We can't all be building our own foundation models from scratch. It just doesn't make sense as an ecosystem. So it's important to have these open platforms so that we can interact pre-competitively, build the best foundation models, and then from there we can get into federated learning, which builds on top of the foundation models. We can then get into fine tuning on our specific data, which builds on the federated improved models, and ultimately, drive our programs forward. So the OpenFold Consortium is a group of over 40 biotech and pharma and tech bio companies involved in this initiative. We have internal engineers and compute, but we primarily work with academic leaders in the field who are driving the innovation, and then we work with them to essentially package, deploy, and make these tools accessible, scalable, et cetera. And so we have 3 programs right now. One of them is OpenFold itself, which we have the honor of having Mohammed here, who is the PI leading this whole initiative from Columbia. So he'll take us through a tour of what OpenFold is and the broader co-folding space. Gabe Rocklin is generating data, mega stability data, millions of data points in the lab. And so one of the pieces that we all talk about is we need more data, but how do you generate those data? And one of the pieces that's missing from a lot of literature data is negative data. And so it's not just about the papers and the stuff that worked, it's a lot about the stuff that doesn't work. And so we're generating that there. And recently the Institute for Protein Design, led by David Baker, has joined. And so this OpenFold initiative is really just starting to take off. And with that, I want to bring up Mohammed to tell us a little bit of the background, past, present, and where things are going with OpenFold. Thank you.
All right, it's a pleasure to be here back at BioIT. I hope to just sort of set the stage a little bit by giving a bit of a perspective on where this space has been in the last 5 years and where it's going, I think, in the next 5 years. So this will be sort of a broad overview of the science of these foundation models and what they're enabling us to do today. I would say it all sort of started with this problem, right? The problem of taking a protein sequence and trying to predict its structure. This is a problem that's been with us for many decades, and it was sort of this kind of foundational really problem in biochemistry. But suffice it to say that for much of the 2010s, there was very little progress in this space. And this was sort of quantified by these competitions called CASP that happened every 2 years, and that invited labs from all over the world, industry and academic, to make predictions of protein structures in a blind way. And so it really challenged these systems to behave without known structures. And what we typically saw during those years is that these predictions were very poor. So this is sort of quantified by the y-axis, which quantifies the RMSD, the root mean squared deviation of these predictions, and it was typically very poor. What happened almost miraculously over the next 4 years was the introduction first of AlphaFold and then AlphaFold 2, which saw these very dramatic advances, essentially compressing really decades of progress in about 4 years, much of it driven by this deep learning revolution that we're sort of all now living through. So as the result of this was really the development of this sort of bespoke architecture, this system that was custom designed for tackling the problem of protein structure prediction. So this was one of those instances where AI and science really combined to tackle a problem that was up to that point not possible. Now, at the time, AlphaFold2 did solve a very critical problem, but it also had limitations, right? So on one level, it was able to predict structures of proteins very reliably. You could give it a protein of up to, say, 2,000 residues even, and you get a structure back that is on the order of about 2 angstrom in accuracy, in coordinate accuracy. And this is sort of an illustration of that, where the ground truth crystallographic structures are in green and the AlphaFold2 predictions are in blue, and you see that they're highly correlated. There were already some sort of funny properties that these systems had. So for instance, in this one particular example, you had an active site that was coordinated by a zinc ion. And in this formulation of AlphaFold, this AlphaFold2 system, ions did not exist. It did not really have an understanding of anything other than just protein structure and protein amino acids. So the fact that we're sort of able to still produce this well-coordinated active site indicated some measure of hallucination, if you may. Right? This model was sort of imagining there's something there, even though it doesn't quite understand what that thing is, being the zinc ion here. And you could argue that this was sort of either a bug or a feature. It was able to predict maybe what you want, but it did not provide sufficient control over the chemical environment of the prediction. There were other limitations. It sort of struggled with multi-domain proteins, and in general, it couldn't really handle complexes, ligands, cofactors, and things of that sort. So this was AlphaFold2, but it was obviously, nonetheless a huge breakthrough. Ultimately, it was recognized by the Nobel Prize a couple of years ago. And one critical thing I think it did that was quite different from preceding systems is that it provided calibrated predictions. So it not only gave you an answer, but it gave you a sort of a confidence in that answer. And this sort of correlation here essentially indicates how well calibrated the system is, comparing the predicted accuracy with the actual accuracies. And the reason why I say this was very critical is because it sort of enabled biologists to now look at these predicted structures, and if the structure that they're looking at is largely blue, that indicates high accuracy, high predicted accuracy rather, and if the structure is in orange or yellow, it suggests low predicted accuracy. So the model is not very confident in those regions. And this calibration really enabled, I would argue, AlphaFold2 to be widely sort of uptaken by the community at large, because it became very easy to essentially ground one's intuition and one's inferences of a biological molecule in terms of the confidence that is presented by AlphaFold. And this was, I think, a sort of a key aspect of the breakthrough. And as a result of this, AlphaFold and DeepMind more broadly were able to sort of now apply the system at sort of proteome scale to the human proteome and ultimately to essentially every known protein in existence to really sort of considerably expand our understanding of protein structure space. So that was kind of Act 1, if you may, from this area. But obviously, protein structure is only one ingredient in a much more complex system that we typically want to understand. So then came AlphaFold3, which is a more general biomolecular structure prediction system. This is a system that not just predicts the structures, but predicts nucleic acids, small molecules, carbohydrates, and what have you. And that yet introduced a new architecture that is even more bespoke and even more specialized for the task at hand. So this is an area, again, where we see really specialization being key to driving progress in this space. And AlphaFold3, in a way, pushed the frontier in a perhaps not as qualitative a way as AlphaFold2 did, but nonetheless sort of opened up this new space to machine learning and to deep learning to become really sort of first-class citizens. And so this sort of figure, he kind of summarizes performance on the left-hand side on the problem of predicting small molecules bound to proteins. The AlphaFold predictions are generally in blue, and sort of relative to kind of the existing systems in gray, you see pretty significant gains. Similarly with nucleic acids in the middle, and then finally with sort of protein complexes on the right. And maybe the most notable thing here was antibody-antigen complexes, which is obviously of therapeutic relevance, where the light AlphaFold3 shows significant gains over the dark blue AlphaFold2. Right, so there's relative to the previous incarnation, you see very significant gains. So this is all great and well, and furthermore, not only are you able to make predictions, but it also sort of demonstrated this principle of inference time scaling that we now see in, say, large language models that essentially corresponds to the fact that with additional compute, you are able to basically improve the accuracy of your prediction. So what these figures are now showing is essentially the accuracy of antibody-antigen complexes as compute is scaled up on the x-axis. And with AlphaFold3, again in light blue, you see an increase in accuracy as more compute is put in, relative to, say, AlphaFold2, the previous system, where there was this kind of saturation effect that doesn't actually benefit from additional compute. And so this is all very exciting, of course, because it suggests that, merely with more compute, we're able to actually produce better results. So that's all fine and well, but are there any things that AlphaFold3 can do? And the answer, of course, is that yes, there are many things that it cannot do. So just to give you a sense of that, going back to this very figure that I just showed you, for small molecules, for the kind of this core problem in drug discovery, even though the results looked impressive relative to the previous state of the art, the way they were assessed was using this kind of post-Butler's test set or benchmark, which is fairly easy, let's say. So it didn't really truly reflect what a drug discovery program would actually encounter in practice. Similarly, for nucleic acids, while you see there's a delta in the bar chart, the fact of the matter is that the absolute accuracy still remains very poor. So we're not really able, I would say, in general, to predict complexes involving nucleic acids, particularly novel complexes that are structurally unfamiliar with any measure of true accuracy. And then coming back to this problem of antibody-antigen complexes, where I said there was this sort of exciting development with sort of inference time scaling, the fact of the matter remains that the compute requirements are really quite substantial. And even then, the accuracy that you ultimately get at from these systems is still quite poor, right? So this remains a frontier both on the kind of the speed as well as the accuracy front. And just to give us a little bit of sense of this is a figure that's often shown in academic conferences that quantifies the ability of AlphaFold3 and related systems to make predictions as a function of the target model, or the target molecule rather, to the training set. So essentially on the x-axis, one is quantifying how similar is the target prediction to the training set that the model has been trained on. And as you get closer to the training set, moving rightward, you see that the predictions get more and more accurate, suggesting that the model is able to generalize in regimes that are familiar to it, but is less able to make reliable prediction as you go further away from that regime. Similarly, these models have limited ability to capture conformational changes. So if there's a molecule that induces, say, a new conformation in the structure, that's not really reflected in these predictions. And this obviously becomes a major bottleneck in being able to reliably, say, model allosteric modulators or cryptic pockets or similar types of systems. So this is where we sort of began to come in, both to address the kind of technical limitations of AlphaFold3, but also the licensing limitations. And the system was not released in a manner that sort of allowed people to either build on it, particularly in the commercial space, or to extend it and use it internally. So there was very severe restrictions on that. Woody alluded to this OpenFold initiative that I had co-founded a few years earlier with several industry partners, and the kind of it's really a sort of a two-pronged thing. Partly it's a code base. It's essentially a set of tools that allow you to build these types of models and extend them and apply them. And it's also an academic industry consortium that provides a steerable mechanism for industry to support science that is open source and that's broadly useful, but it's also in tune with the needs of industry. And just to say here that's the kind of the current generations of OpenFold are very highly performant. So when you look at these sort of assessments, again, by difficulty, you see that it is comparable to AlphaFold3 in terms of accuracy. And equally critically, it's just fully open. So all the code is available, including the training code. All the data is available, including the training data, which means that these systems, or that this particular system rather, is fully reproducible. Right? Provides a recipe that anyone can use to actually reproduce it and build on it, which is not true of other systems in the space. So I'll end with just the next 5 years and where I see this is going. I think we'll see improved generalization by sort of better understanding of the underlying chemistry and protein energetics. I think that's going to be key. I think we'll see improved scales, being able to tackle larger complexes and finer resolution, so that we're able to sort of more carefully capture the atomic coordination involved in interactions. And finally, speed. It becomes quite critical, particularly when we think about applications like virtual screening, where we would like to make predictions really at the scale of billions of potential molecules, which is not currently tractable with these systems. Thanks for your attention.
Good morning. Thank you, Mohammed, for a great introduction to thinking about foundation models. And now I want to talk about, as we take these foundation models, how do we move them from training on these open data sets to moving into the proprietary data sets, which I think many of us have in our own databases, and how do we use distributed private data sets to improve model performance. And to start this off, actually, my colleague JT had a great slide in his deck that I pulled into mine, and this really reflects the idea that at its core, biopharma takes investment, through as a smaller company taking investment, and turns that money into proprietary data. That data is really not meant to be shared. And as a result, you have these barriers, these walls, these silos that are put up between companies, really blocking the improvement of models. And really this points to the need for federated learning. How can we improve models while maintaining the privacy of the underlying data sets? And so that's what I'm going to talk about, giving it a brief introduction to federated learning, then talk about how Liully has taken this forward in developing ways to collaborate and improve model predictions for things like ADME tox, but also continue to expand and push that barrier in protein to design, protein predictions. So first, what is federated learning? As I mentioned, it's not surprising that companies are very sensitive to the proprietary data that they've spent incredible investments generating. And so federated learning was generated in response to that need of how can we improve model performance while maintaining the privacy of the individual training sets. And so at its core, you have a global model as a particular architecture, say it could be OpenFold 3. And in this model, you have an initial set of weights and parameters. And then you have different parties that have proprietary data sets that are stored in separate environments. They're not exposed to each other. All that data set is maintained in its privacy to that company. Critically, the model goes to the data. So the model is then moved into that environment, trains on that data, say in that top red box, training on that data. Any changes in model weights or gradients are then integrated into that local model. And you can repeat that across, here I'm showing 3, but you can imagine this across some of the work that we're doing in dozens of companies, providing individual private data sets. That those model weights and gradients are then brought into a federated learning server over on the far right of your screen, where they're aggregated. And then those modified weights are then brought back into that global model. So that's one cycle of this process of the model moving to the data, training, bringing back into a global model. And then you can imagine the cycles repeating multiple times as that model is then updated, brought back into those environments. Those model weights are then adjusted again. And you're repeating this cycle until you reach an optimal or until there's certain cycle sequence is met. And I think this idea then allows this model to improve while still maintaining the privacy of the individual data sets. Those are never exposed to any of the other parties, never exposed outside of their private environment. And so this general concept really opened up the idea of how can we collaborate across all these proprietary siloed data sets. And that's really where TuneLab was born out of. So Liully created this effort, which is a first-in-kind effort, a collaborative AI/ML effort between Liully and biotech and the biopharma industry more broadly. And what it's providing is Liully's AI/ML models provided to biotechs to use them at zero cost. These are the same models that we use every day. These are models that have been trained on decades of internal data sets. It's estimated to be over $1 billion of data that have been brought into the models by Liully. And as part of joining TuneLab, biotechs are able to use these models, but are also expected to contribute data sets to help improve it. And critically, building upon the infrastructure of federated learning, and which has now been in use for over a decade in many different industries, allows the models to improve without exposing that underlying data set and preserves the privacy of that data set. And this was a first-in-kind effort that launched late last year. We've seen an incredible excitement about this. And so, launched late last year, we had a great uptake, great industry excitement at JPM, a great number of references. And In these past 6 months, we've exploded in the kind of usage and excitement around this. We have over 75 partners in TuneLab. It's been approved across 3 different continents and dozens of countries in use, and we have partners in almost all of those jurisdictions. We see a breadth of, in our case, focusing on 2 specific areas. We have work in small molecule as well as antibodies broadly. And so we've seen in the small molecule space just a breadth of different kind of molecular approaches, from things that are Rule of 5 compliant to things that are well beyond that. And we're seeing value, and our partners are seeing value in making decisions, data-driven decisions in areas that are challenging to do. And I'd put a number of different partners that have joined, and we're really excited about the different types of partners that are joining and the different stages, where you can have companies that are obviously not listed here that are in stealth, that are making their first program, moving it towards the clinic. You can also have companies that are commercial stage, who are public, and are developing multiple programs. And I think this really reflects the value that the idea of having access to transformative models to help speed up the drug discovery process. And I think another aspect that I think is important to think about is just realizing that the model is the criticality of allowing a model to both be improving its performance, but also making it simple and easy to use. So we spent a lot of time building out an infrastructure and an interface that allows partners to use these models in a simple way. And so right now, these partners are logging into a website where they can make predictions on a whole variety of different characteristics. As I mentioned, right now, we're focused on small molecule ADME tox and antibody developability. These are models available right now. There are, I think, about 40 models and more coming soon. We've been receiving feedback about what models to add and working with partners to bring in those models, provide them to the community. I think it's been a very successful feedback. But I think we also want to think about how can we meet scientists where they are. Scientists are working in different environments as they're designing their molecules, as they're storing their molecules. And so we've worked with a number of different partners to provide access of TuneLab members in their local environments. And actually, this morning, I'm excited to announce that as of 8 a.m. this morning, we announced a collaboration with CDD Vault, which a number of partners have asked for, is how can we bring models into CDD Vault? So they're our newest kind of partner to allow access to models into these environments. And so we're working on bringing these models into those spaces, including into benchling, Schrodinger, Revity, and allowing things through API access. And so this is an active work in progress, and thinking, how can we scale this? How can we reach hundreds and hundreds of biotechs to allow them to have access to the greatest models, allow them to support and build a community to improve those models such that we can all create medicines faster for more people. If you're interested, please reach out to us. There's a web form. You may think it goes nowhere. It actually goes to my inbox. So please find me afterwards or fill that out. We'd love to chat with you and identify if you're able to join TuneLab. Hopefully that was helpful on a little bit background on federated learning, how Lilly's applying it, how we feel that it can continue to accelerate drug discovery and bring more drugs and therapies to patients. Now I'm going to pass it off to my colleague JT, who's going to take us through a little bit more about how do we move this additional information on federated learning and fine-tuning.
Thank you, Jonathan. Great news and exciting announcement as well. I think that as I'm thinking of federated learning, I like to give like a very easy comparative like metaphor to an extent. I think of federated learning as a techno-regulation kind of solution, and think of seatbelts, car seatbelts, right? So we use seatbelts, but sometimes they're very uncomfortable, so we take them off. But it's really difficult to drive with your seatbelt taken off because you have all of these lights popping up everywhere. You have sound, boop, boop, boop. So it's really difficult to be to have a bad behavior. And federated learning is kind of the same in the sense that it protects. It makes it very difficult to behave badly when you're looking at very sensitive, sometimes secret, proprietary data. So that's sometimes I think of it like that, and hopefully that helps for those who had never heard the term before. Now, what I will talk to you about is how it takes, what it takes to actually move federated learning to industrial setups. I will talk to you about this for the next 10 minutes because I will make the link to what Woody was saying before around how to use fine-tuned models as well. So at Apheris, we power the largest federated networks for drug discovery. And we do this through our products. Through our products, companies can join or build their own federated learning networks. And with these products, we actually protect the most important assets in your companies, which is the sensitive data, sometimes the secret data, as I said before. In addition to that, Apheris also helps setting up the infrastructure so that these sometimes very demanding workflows actually can execute in your environments. And these are some examples of the networks that we power. We have the AISB network, where we have 9 top 20 pharmas. We're focusing on co-folding. We're focusing on protein-protein interactions. We're focusing on binding affinity. And there are other networks as well. We power, for instance, this ADMET network, where we're focusing on small molecule property prediction. We're now co-driving the antibody developability network with Ginkgo DataWorks. And for this, we're building a purpose-built data set to kind of optimize model development. Importantly, across all of this, and we will be soon launching a binding and structure prediction network, more of this soon. But I guess the common denominator for these networks are that the models are trained on proprietary data in a secure way. So the data is never leaving the environments of any of these nodes in the federated network. And the other important point is that all of these models are delivered to the nodes, to the different companies. So the companies can take those models and do what they need to do. They take it in their internal programs and use them however they have to use them. Mohammed talked about OpenFold 3 and some of the other foundation models. And one of the most important question is, what do you train them on? So John stole my little slide here, and I take that as a compliment. And it's obvious that there's a lot of public data, but the public data kind of skews towards well-characterized targets. And the industry data complements that view with more diverse data and sometimes higher quality data. It's not always the case, but sometimes with higher quality data. And if there is something to learn about the AI world today, is that you cannot necessarily model your way out of a data problem. And you can't buy this data either. This is, as one of our stakeholders in one of these top pharmas was saying, that data is our currency. This is not for sale. We cannot buy this data, and this data is absolutely not for sale. So the elegant solution is this techno regulation solution, federated learning, this seat belt, so that this collaboration can actually happen. I think it's quite remarkable that a couple of years ago, all of this was kind of an IT decision from the different companies that are well represented here. It was mostly IT people making the decision of whether to use federated learning products. That's not the case today. Today we have the business leaders trying to get a hold of this technology and to leverage the power of it. Woody alluded to this, I think, for a second or two, but this is not a plug-and-play capability, and we have to be honest about it. Many networks in the past and some current are probably doomed to fail because they're not considering some very special themes around what needs to be done right. And the first one is engineering rigor. Just to give you a sense of what we did for the AISB network, so we trained continuously for around a month with, it was 80 H100 GPUs spread out evenly across the different companies. And imagine all the complexity that comes with that. Each one of these companies have their own network constraints. They have their own firewall rules. They have their own compute windows that they have to negotiate with the cloud providers to make sure that the compute comes online at the right time. They all have their own particular approval processes. And you have to actually do this right, so that with the right federated learning partner to make sure that this can actually happen. The other piece that needs to be considered is that data preparation without centralization is a new paradigm. There's no way of inspecting the data in a central location because data is never shared. And imagine the complexity of this, because each company has, you have your own ways of organizing the data, of harmonizing your data. You have your own standards. But at the same time, you have to have comparable training setups so that foundation models can actually learn from this. So this implies immediately that you have to have tools, you have to have the proper tools for harmonization in this new setup where no one externally can actually look at your data. And all of this leads to, like, you have to have an enterprise deployment with no friction, in the sense that, great, this technology has proven to work, but your IT, your cloud engineers cannot actually spend a month just trying to set it up. So your federated learning partner has to be able to work with your processes, has to be able to understand how to streamline the reviews, the security, the privacy requirements, and all of that, and have actual thread model capabilities so that your requirements are satisfied and that the projects and the initiatives can move forward. And all of this, I'm saying this as it was, it's difficult, but all of these networks, actually, the AISB network is one example where live, this is working, and it's expanding to even more complex workloads. So does this actually work? Indeed, it works. So what you see here is a summary of the AISB 1 project, which is very cleverly the first project of the AISB network. So that's not very well. We also called it in the public the Federated OpenFlow 3 initiative to make it a little bit more understandable. And what you see on the far right here is that is the performance of the federated model compared to other models, compared to Boltz 2, Protenex, compared to vanilla OpenFlow 3 Preview 2. And you can see the huge uplift between OpenFlow Preview 2 to the federated learning model at the end of the project that learned from the data from all of these companies. And this huge uplift is driven mostly, if not exclusively, by the breadth of this new data. Interesting, the second best model is OpenFlow Preview 2, trained on additional public data in an extended cut of data, which proves that that data is actually the differentiator here. We also looked at how all of this compares to the fine-tuning processes, right? And what you see here in the middle is what you see here is the comparison between federated models to fine-tuned models in pharma environments to the vanilla OpenFlow 3 baseline. And fine-tuning sits in between, right? It's not able to, no single company can actually get to the level of generalization that the others, like a federated model, can. So this is kind of the very first, my very first mention of fine-tuning models. And I think that you heard Woody say that we look at this as a potential stepwise process. We actually look at it as a cycle where you have a federated model, then you can fine-tune those models. You can then go back to a federated model and keep iterating on this depending on your needs. And so we actually see, of course, the models will be the starting point, but federated models can also be the starting model, the starting step, in the sense that the federated model will be trained across industry data, constantly improving with new partners joining the networks. But the reality is that the federated network does not know all of your targets, right? They don't know the most recent data that you're working with, the specific program data. And this gap can actually be filled with fine-tuning. If you allow pharma companies and all of your companies you're representing here to actually have the capabilities to fine-tune those federated models on program data, and especially when there's more data coming up from all of these programs. I like to think of this as a benchmarking culture where you're actually constantly evaluating what you're producing internally and see how powerful or meaningful they are. So, and follow me here. So you should see, I think, federated models as a portfolio asset, and then the co-folding models, including these federated models, right, can be fine-tuned, especially as new data arrives. So that way, the federated models have the breadth, but at the same time, they have the local understanding of what your actual current, most recent programs are producing in terms of data. And this is key, right? Because it enables key decisions in lead optimizations, in virtual screening, or in off-target assessment. And we're doing this. I'd like to think that we're democratizing this fine-tuning and benchmarking capabilities with the companies that we work for. Now, in this fine-tuning journey, and I will soon end my little talk here, this is what we did with SandboxAQ. We integrated SandboxAQ Affinity Head on OpenFold 3 within Apheris Fold, one of our products. And we built a data set of 69 macrocycle compounds. We trained on 49, and then we used the remaining 20 as the held out data set to validate what was going on. The predictions improved drastically. The actual validation Pearson more than tripled for this held out data set. And then we took this further. We extended the affinity head to handle ternary complexes like molecular glues to feed signal back into the structure model. We did other things, but at the end, we looked at the results and we could see that performance can be boosted through a series of things, through extensions of the SandboxAQ affinity head, with the OpenFold 3 inference pipeline, but also with affinity fine-tuning and with structure fine-tuning. And all of this gives us the best of two worlds, because federated models, they will give you that generalization by considering diverse chemotypes, but at the same time, fine-tune gives you that program-specific specialization that many of your teams or yourself need. So I guess I will close here. The key takeaways are that proprietary data drives model performance, and federation is how you can access it in this new AI world where you have to protect the confidentiality, your IP, the confidentiality of your data. We showed that federated models outperform public and fine-tuned models. And done right, federation is a standing capability, not a one-time project. So you should not see this as a one thing, but more like a repeatable operational model that through deployments, through governance mechanisms, can keep going in your internal operations. And finally, local fine-tuning is what converts that model performance into drug program impact. And I think that's what over time compounds. And yeah, thank you. Thank you. I will now hand over to my colleague, Armin.
Well, thank you so much, JT, for the amazing presentation and for using SandboxAQ models and fine-tuning them and putting it into action. I love to tell you more about fine-tuning on these models today, but first I wanted to share a little bit more about SandboxAQ. I wanted to introduce what Sandbox is to those of you who might not have heard about us. And then I have a big announcement at the end of my presentation, so please keep your phones ready for scanning the QR code. But going back to Sandbox. Sandbox is an AI and advanced computing company spun out of Google a few years ago. We raised $1 billion to build AI for scientific discovery, what we call large quantitative models, to contrast it against large language models. So as opposed to training these models on Wikipedia and Reddit and images of cats and dogs, we train them on physics and chemistry and biology, as you can imagine. We already use these models in 20+ discovery programs with large pharma, spanning various disease areas and also drug modalities. A team of 250+ scientists and engineers, and growing really fast. So when we say large quantitative models, I wanted to emphasize that we are not really talking about a single model that solves all problems. These are a collection, a suite of models that span the entire drug discovery spectrum, from target ID all the way to early clinical development. This is a suite of specific models for each task. And We have been working very closely with NVIDIA to build these models and to release these models. As you know, one of our key value propositions is that we know how to push GPUs to their limits and to make them go brrr and make noises. And in collaboration with NVIDIA, we have released some of these models recently for the community. We had the pleasure of working with Mohammed and Woody on the OpenFold models, co-folding models, as well as our binding affinity prediction and a number of data sets at the frontier of structure-aware drug discovery. And we are really living in exciting times when it comes to ML accelerated drug discovery. As we learned from Mohammed's beautiful presentation and John and JT, there's really an explosion of new models. We see like every day there's 1 or 2 new models for drug discovery every day. And what we see at Sandbox with our partners in large pharma and various biotech companies is that it's getting a little bit overwhelming and harder to put these models into good use. We see that a lot of these models are amazing in achieving great results on benchmarks, and it's fascinating for publishing papers, but they fail to generalize to real drug discovery use cases. When you are thinking about a serious drug discovery program, it's quite hard to make the connection. And what we have learned the hard way at Sandbox, and we are doing this with our partners, is that fine-tuning could help a lot to bridge this gap. When we are able to, and we showed that in a couple of publications, others have shown this in the community, that even a small fine-tuning effort can dramatically change the predictive accuracy of these models when we are looking at a project, a specific workflow. And really what's really interesting to me is that fine-tuning is kind of like an application of federated learning that federated learning wasn't originally designed for. It kind of reminds me of Listerine, which was first marketed as a floor cleaner before people figured out that there's actually a better, bigger market in oral hygiene and as a mouthwash, basically. So this is, I think we are seeing a very similar moment with the fine-tuning, that the additional advantage that we get using the federated learning infrastructure that is already in place for securely introducing additional proprietary data to a model. And I think over the next few years, we're going to see more and more of these federated platforms being used for both pooling data, but also for fine-tuning these models on project-specific data. And I want to give you an example of this. Johnson & Johnson, together with Apheris, my good colleague, JT, they worked on basically adopting one of our models that we recently released, the AQ affinity model, which is a binding affinity prediction model, and adopted and fine-tuning on a data set. And they were able to show that the error in prediction accuracy can be decreased by 70%, which is a dramatic increase in accuracy. And you can actually start to see correct rank ordering using this binding affinity prediction model for that project-specific data. And like these projects are often hard to drug targets, they are complex, so it's really hard to make the generalization without fine-tuning. And it was really interesting to see that even a small fine-tuning effort can do wonders in terms of really unleashing the power of these models, which don't really work for serious applications off the shelf, and really put it into use and make it practical for a real drug discovery workflow. But the second takeaway, which is equally important or even more important, is what I said earlier, which is the secure infrastructure that federated learning is providing us to be able to bring proprietary data is actually making it super easy to engage in this fine-tuning task. And I think JT covered this very well in terms of like thinking of it not as a sequence, but as a continuous flow of like federated learning and fine-tuning. And I think this is an application that we're going to see more and more of. And as you can imagine, with this effort and what Mohammed presented earlier, we are constantly at Sandbox thinking about how to make these models more accessible. I mean, with federated learning, the team at Apheris and a lot of other groups are basically solving one of these problems of basically being able to pool the data together. Now we are thinking, how can we make it even easier and more accessible, these models for everyone? Because we believe that drug discovery is hard. And the more smart people we have thinking about solving these problems, the better chance we're going to have as humanity to be able to design better therapies faster. And today, I want to make a big announcement. But before that, let me actually, because this is a morning session, let me check how many people are actually awake and paying attention. Quick question. How many of you are using large language models on a daily basis in your daily life? Maybe with a show of hand. Amazing. Well, as expected, everyone. There were a few hands down there, but I think they are asleep. So now the follow-up question to that, how many of you are trying to use large language models for serious drug discovery workflows, for doing your technical work? Great. Well, a few people, but not the entire audience. And this is what we are trying to solve at Sandbox as one of our go-to-market motions, is to connect our large quantitative models to large language models, so you can, in an agentic framework, you can access all these different large quantitative models, all these sophisticated drug discovery machine learning models, and run complicated drug discovery workflows just by talking to your large language model in plain English. No code required. So with that, our marketing team has created a beautiful short video, and I want to show that to you if it works. Let's see if the AV works. Yes.
You can now discover new drug disease insights or simulate chemical catalysis reactions without writing a single line of code. LLMs can now access physics and chemistry-based models, exposing complex mathematics and simulations relying on matrix calculations. Until now, access to these models has required setting up virtual machines, tuning performance, maintaining infrastructure, not to mention command line coding. In contrast, here's the same model accessed through an LLM. No virtual machines, no setup or coding required. Same rigorous science, but faster, easier, and more accessible. This is just the beginning. Learn more about what's next at SandboxAQ.com.
Amazing. So I'm going to finish that by saying we have announced this week, and we are getting an overwhelming response date. So we had to create a waiting list to slowly roll out the model. And as of today, the waiting list is live. I have the QR code here. Feel free to scan it and join the waiting list to get access to these models. And if you couldn't get there, just come talk to me, or my colleagues are here as well, so you can sign up to get access to these models. And with that, I want to thank you for your attention and call Christine to the stage to tell us more about now the applications of this in real drug discovery workflows.
All right, thank you very much. So I am I'm the computational molecular design lead and also senior science fellow at Bayer Crop Science in St. Louis, Missouri. And I'm going to talk to you a little bit about how we've been leveraging some of the tools, especially as far as foundational models and biomolecular AI, to drive decisions at Bayer in both crop science and pharma. First, though, I'm going to talk about the other hat I wear. I'm a member of the OpenFold executive team, and Bayer is a member of the consortium. And I'm really going to talk about some of the community impact that this has had to help us drive some of our research forward at Bayer, and opportunities that everyone potentially has if they were to join the consortium. So really, I think the community-driven aspect is very important, particularly with what we saw, as far as driving things eventually towards maybe like federated learning and that sort of space. But really, I think that this community-driven software has really allowed faster innovation in the field overall. And really, by sharing some of these foundational architectures, allows everyone to be able to drive biomolecular AI forward, and really has driven some of the very quick advancements we've seen in the field over the past few years. In addition, I think Woody mentioned this as well, but really from a sustainability standpoint, everybody doesn't need to be training their own foundational models. Really getting together in a pre-competitive sense to develop these foundational models from which we can build upon is really going to be a key area which is going to drive innovation. The really interesting thing about being part of the open fold ecosystem really is enabling the community to drive where the field is going. These are scientists, particularly like me at Bayer, as well as others, that are really determining what the aspects are that need to be developed to drive the field forward. What is industry really needing? Where are the gaps? And putting effort in that area versus, from a purely academic sense. So basically, we have a very fast translation from, implementation to being able to use it in industry. And some of the areas that we're interested in as far as community development across is really trying to drive things like, for instance, training on waters. We know those are important, but none of these biomolecular tools really have implemented that. So currently that's a very exciting, interesting area that we're focusing on, as well as antigen modeling, and also protein design. In addition, we all know that experimental data is incredibly important for improving these models, and the consortium is investing in that as well. And the community aspect is also very compelling. We have a technical advisory committee where many people from different software companies and biomolecular pharmaceutical companies and ag are coming together to really solve problems in a pre-competitive sense, and building that trust, which will help us move the field forward. All right, now particularly at Bayer, I'm going to talk about some of the ways really that we've been able to use this, as well as speaking for other folks in the consortium as well, how these biomolecular tools have really advanced and how we've been putting this into a framework that we can drive decisions and drive faster product development across the pharmaceutical space, as well as the agro-science area. One very obvious aspect really is using these tools to solve crystal structures better. One of the big problems with solving crystal structures is actually determining the phase. And by doing the protein folding, we're able to actually solve these structures faster and more efficiently. Another thing is taking these foundational models and fine-tuning them, as was mentioned before. We're using that quite regularly to improve our development of biomolecular pharmaceuticals, as well as some of our crop science traits for development of herbicide tolerance traits, insect control traits, et cetera. Also using some of these tools for protein design to develop new traits and also new biopharmaceuticals. In addition, we've been using these for drug design, as well as the crop science development that I mentioned, and also leveraging the tools to look at protein-protein interactions. We've been also using this in terms of using this for mechanistic modeling for like enzyme catalysis or biotransformation. And we also are leveraging models and fine-tuned models to do protein stability, solubility, and also expression. And these foundational models have really been critical to be able to drive this forward. However, we use a variety of tools, and it's not just one tool really that is helping us to solve these problems. So There's also been, a great deal of advancement over the past couple of years, as you've seen in this biomolecular space, but by far the problems are not solved. It's not a turnkey push button solution. And now I'm going to pass it over to Woody, who's going to talk about some of the grand challenges that still exist in this biomolecular AI space.
Great, thanks, Christy. So I promise we'll get to the panel soon. Just wanted to highlight the fact that the stuff that we're all doing is hard, and so you're hearing a lot of the positive stories, and there's a lot of progress, and we're making things better as a field. It's amazing time for all of us, and yet we're really just getting started in terms of using computers to predict what molecules to make next and how biology works. It's enormously complicated. And just as an example, I took on this adventure about 2 years ago to quantify the grand challenges just for small molecule drug discovery, and then surveyed folks in the field and from all of our experiences, and worked with a bunch of great academics and folks in industry, and ultimately came up with a set of 22 grand challenges. And so we've been talking about some areas like protein structure prediction and dynamics, but to make a drug, one has to understand chemistry and structure and energy and all these pharmacological endpoints that are all complicated. And all of this is just for small molecule drug discovery. There's obviously the same types of issues, but different for biologics, all different, antibodies, for example, ADCs, gene therapy, cell therapy, these all have their own grand challenges. And so I just want us to all realize that language models, as have been said, are great for automation and things that can be described with language and orchestration. But we need these, as Armin said, these quantitative models to actually make predictions about the things that matter in the lab. And language is just not great at explaining the physical interaction between molecules in the complex environment of biology. So as a transition, I'm going to invite everybody up here now for the panel. We have a few questions, and so if the panelists can come up, we're going to cover these themes. A lot of it has already been described here, so everybody come up and work your way over here. This is just to give a set of what we want everybody to get out of this by the end. Just as a transition, we're going to be able to cover all these areas in some form or another, but again, this is really just the beginning of the conversation. Again, if folks want to enter questions through Slido, please do that. Ali will be fielding those, and we'll, with that.
The code, which is not on the screen right now, is BIT26, no hyphens on this one, and I'll be taking questions there.
Great. All right. Super. So we already covered a lot of the background questions, and so thank you everybody for getting into that. Wanted to start a little bit by just digging into some of the issues with federated learning and what are some of the biggest. So we talked about the high level, the folks that are doing the federated learning. I want to talk with first the professional organizations and how that's what's coming in and what's not coming in, and what are some of the challenges, not just the technical challenges, but maybe some of the legal challenges and some of the kind of people, social challenges of getting folks on board. And if we're in a situation where maybe the best data are still being reserved, maybe that's where fine tuning comes in. So maybe start with there, and then I want to migrate over to Mohammed to talk about that same question, kind of outside of this private world of data, and how we can start to leverage this more broadly for the entire community. So maybe John and JT can start with that, and then we can open it up from there.
I think JT made a good point, as the understanding and the impact of federated learning is growing. I think it's moving from a more of an IT discussion to there's a business need, there's a driver from the scientific side. So I think that's helping overcome the challenges. I think there's historically been a challenge of just a nervousness of taking proprietary data and allowing a model to access this even in a privacy preserving federated way. And so I think now that you're driving from the scientific side, seeing the gains in performance, I think we're overcoming that. And as JT and we're seeing, there's a growing comfort with this idea, and people are seeing benefits. So I think we're well into the progress of implementing this as a regular methodology. That's just a standing capability.
Great. Could not agree more. And I would probably categorize the challenges. There's the technical and different flavors of technical. There's on the infrastructure side, there's on the data preparation side, and there's others. I would also categorize the organizational problems. Like the organization entirely needs to move towards a pace of collaboration, and that not always is the case, that it's an easy thing to do, right? And then there's the legal challenges, the contractual challenges. This introduces new ways of collaboration, things that the contract expert, the legal experts are not necessarily always used to. And in the world of AI governance, well, they're starting to get used to it, because this is something that the business leaders, the scientific leaders are pushing for. So in all of these dimensions, technical, organizational, and legal, little by little, we're finding how to optimize the way to kickstart an initiative, but at the same time, make it repeatable. And I think that's one of the most exciting pieces of development in the last year or so. And hopefully this will continue to evolve positively. So, yeah.
Great. So, there's kind of this pay-to-play thing that's going on right now, which is great, and folks are contributing, and folks that have data can get access to these. Folks like Lilly are actually opening up more broadly. But what about the rest of the ecosystem, the biotechs, the academics, everybody else who doesn't have the data and maybe the financial means to contribute? How can we think about broader federated learning, or is there a place for broader federated learning, or is it really just working with public data? So maybe Mohammed, some thoughts on where this is going to go across the entire ecosystem.
Yeah, absolutely. So I do think federated learning is here to stay. So I think even as these models get better and better, and as we have broader public data sets, I think there's going to always be sort of a gain to be had, essentially, from pulling data from private pharma repositories. One thing we've been involved in, however, to sort of try to kind of lift the baseline that we're all working from, is this open bind effort, which is really targeted at essentially generating data at scale in a publicly sort of facing way. And in an intentional way, so that the effort is focused on essentially improving, broadly speaking, how these models behave, and not necessarily targeted on any one given target space or chemical modality. So I think there's synergy there between kind of the public space generating sort of general purpose data that lifts that baseline, and then of course company data coming in and really specializing these models for more difficult modalities and ones that are that have high value.
Great. So Armin, maybe you can comment a little bit, since you guys are kind of spanning the spectrum. How do you envision folks providing their data through federated models versus the fine tuning? We kind of went back and forth talking about these two concepts. Where is the line? Are people going to contribute all their data for federated learning? Is there going to, and what kind of data is going to go in? Is it going to be kind of the old stale data, which is maybe useful, but maybe not as useful as the fine tuning? Where are those lines drawn between where people are going to contribute and where people are going to keep it proprietary and maybe use for fine tuning?
Yeah, absolutely. That's an excellent question. So to me, what we have seen in the market is that not all data is generated equally. So there's different quality of data, and there are different applications where in some areas we see there's more data scarcity problem, and there's a greater value in being able to pull different data sources to be able to train models that are more generalizable. So certainly there are areas where pulling the data together is more valuable than others, and not all data is generated equally. But what I want to focus on is actually the infrastructure that powers federated learning. I don't want to think of this as federated versus fine-tuning, because on the surface it might appear that these are actually two opposite paradigms, because one basically involves pulling the data together, and then another one is more focused on keeping the data and kind of like make a customized and fine-tuned version of the model. But actually, these two on the surface opposite paradigms are actually very related, and they have the federated learning framework common between them, because the secure infrastructure that has been built to allow for handling proprietary data coming in and in a secure way being federated into a larger model. It turns out it's actually the exact right tool that we need to facilitate fine-tuning when a company is looking into adopting a new model and bringing a new open model and training it and fine-tuning it. So I loved what JT described earlier as looking at this as a continuous, basically, process where there's federated learning and fine-tuning. And I think that's really where we're going to see the most value, or the compounding value of both these two paradigms working together.
Right, and we're seeing all of this come together, and it's happening, and I think folks are joining because of the FOMO. When you see the data, it's quite compelling. So Christy, maybe talk a little bit about when this all starts happening, and it is happening, and as these models get to be more and more accurate, how does that actually change how you do your work, and how the field as a whole is doing work to design therapeutics, to design better crops, just to improve the overall state of what we're all trying to do in humanity here?
I think the big first step actually is having a open consortium, like we mentioned, because within that consortium, we have about 40 companies that are part of the consortium at this point. And really, it's a matter of building trust and working together. And I think that's laying the foundation for doing these next steps, potentially with federated learning. I think that really, as we grease the wheels towards some of the legal implications and that sort of thing, I think that probably additional companies will join. Bayer, for instance, was part of a federated learning aspect called Melody back a number of years ago, and there's a paper out about that. So I think some companies have had experience in this area as well, and I think that especially now with these data-hungry AI models, it's going to become increasingly important.
Yep.
Woody, we've got some questions.
Okay, let's
a bunch of questions actually. So I'm starting with the ones that are upvoted. LLMs can be tricked or jailbroken into regurgitating training data. How do you address concerns about this on coveted private data sets today?
Great question. Maybe with folks doing the federated learning.
You want to take this one first?
Yeah.
Well, that's a great question. I don't have a particular view on LLMs on this specific use cases that we're thinking of. The way we're trying to run federated learning frameworks, and I think it joins one of your questions before on the contribution mechanisms and how parties participate in these federated learning initiatives, is that first and foremost, the data needs to be protected. And that doesn't mean just the core data sitting in your environment. It also means that you cannot reverse engineer the data from the trained models. And at Apheris, it's not necessarily the standard in the industry, but at Apheris, what we do is, as part of all of these initiatives, before releasing any model, we conduct a very rigorous threat model exercise where we go and actually attack the models to see if we can reverse engineer the data, if we can do some memory inference attacks and others, to give the reassurance to the different parties that the model that they are getting is not doing exactly what this person is asking for. Yeah.
Jonathan, anything to add?
In our case, we're working with a partner called Rhino, who's been doing federated computing for, I think, over near a decade with hundreds of companies. And I think this is a general challenge that they've faced in those different industries. And so we're confident in working with them and doing some of these threat analyses as well ourselves to make sure in TuneLab, obviously, we're very sensitive to this, as the large majority of the data set is the 20 years of Lilly's proprietary data that is not public. So ensuring that those models that are out there are not able to be reverse engineered on the data set.
It's a great job opportunities for hackers to get hired to try to hack before it gets hacked.
Is there an appetite to collaborate with the federal government agencies too with these federated models?
Mohammed, you want to maybe jump into some of your thoughts around what the government can or should be doing?
Yeah, I mean, so this sort of relates back to what I was talking about earlier, where I do think there is synergy between the government essentially lifting all boats, right? So it's going after problems that are broadly available and useful to companies across the board, and then companies coming in really with their own specialized modalities. Certainly, I would say, I think with the stuff that we've been doing with OpenFold and like I said, OpenBind, there's been an appetite for federal funding to come in and support these tools. The one challenge I will say, however, is that a lot of the work here is very engineering heavy, right? Both on the federated training side, but also in terms of building these models. And it's very compute heavy. And I will say, at least in the US, the kind of the funding infrastructure is structured in a way that is not maybe all that in tune with kind of the modern needs for these types of systems. And this is in a way where we actually stood up OpenFold in the first place, because we felt like it wasn't being sufficiently sort of understood as a key enabler by the federal agencies. I think that's perhaps changing a little bit, and I think there are now new philanthropic sources that do realize the value of software. The Chan Zuckerberg Initiative, for instance, was one of the first to make that association. But I still think we have a way to go to really get to a sort of a healthy funding ecosystem that spans across pharma, industry, academia, and the government.
How do you balance improving the global model with the risk of another company getting to market first when establishing a federated learning framework? How is this pitched?
Any takers? There's the pre-competitive angle. Rising tide floats all boats, and if you miss out, you're behind. And at the same time, and it kind of comes to the question of, do you contribute your most proprietary data? If the model surges and you get this amazing performance boost one day after some round, and it wasn't your data, it was probably somebody else's data. It gives some insights as to maybe what people are working on. But yeah, maybe a little bit more about how do you distinguish that competitive angle and what is you don't want to share with everybody versus knowing that contributing to the field is going to help you and help everybody, and ultimately make it better for the field.
I wanted to add to this and say, I think the cost of not being part of a federated learning framework is much higher than the risk of being in the federated learning framework, because AI is basically garbage in, garbage out. If you don't have the right data, then maybe you could engineer something that looks good on a publication and for benchmark sets, but in order to put it into action for real drug discovery workflows, you need to have high quality data, and the cost of generating this data and the time that it takes is basically, I think, much higher if you want to do this alone compared to the federated learning process.
I would very much agree with that, Arman. It's an opportunity cost. You evaluate whether to join or not. But the reality is that when companies join all of these initiatives, there's clear incentive mechanisms and there's clear contribution rules, right? Depending on what the network is, there's analysis about the diversity of the data that each partner is contributing to, the novelty that each partner is contributing to, and some things that are privacy preserving to in a continuum of what privacy conservation means. Well, kind of gives that reassurance to the partners that, okay, I'm getting this in and I will get out something that is still meaningful for me. And ultimately, it's I think someone mentioned it. I think you did, and it's a matter of trust, and the trust is not just something in the air. It's trust through the processes, through the legal mechanisms, through the contractuals, through the everything that is going on in the federated learning network. Hopefully, we have answered to an extent that question.
All right, we've got a lot more questions. I'm going to do one more. What kind of data is best for federated learning? Are there data types that are best suited for this application?
I can take a stab at this. I mean, certainly with the AISB1 consortium that JT mentioned, we saw very significant lifts with structural data, and I think in part because chemical space is so large that in a way having different companies almost invariably is going to help, because they're exploring these regions differently. And what we actually see, I think there's sort of two modes. There's the kind of general purpose understanding, rather the ability to generalize, where essentially data from any company typically helps, because again, it's sort of introducing new physics to the problem. And then there's a more memorization, let's say, kind of regime where maybe problems that are very related to their target space get much better, but then there's the limited effect, beyond that regime. But I would certainly say structure, I think, is a very natural one. I think binding data is another. That's something we've been thinking about, both because of the volume that's available and for similar considerations. Chemical space is large, and it's sort of easy to be non-redundant.
And Jonathan, do you want to maybe talk about the consistency of the data and kind of how to make sure the metadata and everything comes together so that you can actually pool all this stuff?
Yeah, I think the quality of the data, but also the metadata, the associated information, making sure that it's understood how the data was generated. From TuneLab perspective, we started with ADME talks in part because there's more standardization and developability characteristics. There's more standardization of how those data are generated through CROs. I think another thing that made me think about your question was thinking about where is there limited data in the overall drug discovery continuum. I think we've been talking more about structural binding information, but something that we've announced and will be launching soon in TuneLab is multi-species in vivo PK modeling, multi-species in vivo tox modeling. Again, that's getting later stage. You've much more limited data sets. There's much more limited information in public settings. And so if we can move into the spaces as you move down from, say, identifying a binder to, will this have good drug-like characteristics? Will this drive toxicity issues? What can you start to predict in human PK? Can you start to predict human tox? Those are some of the areas where they've incredibly limited, incredibly expensive data. Driving federated learning in those spaces could be, I think, a massive lift for the drug discovery industry, and that's something we're working on.
And I think something that Liully TuneLab has done very well is, in order to get access to these models, you have to contribute data, and you basically get a bonus if you contribute data that follows a standardized protocol. And so all data, is good, and better, high quality, more reproducible, consistent data is better. And there's actually a bonus that if you contribute those kind of data, you essentially have to contribute less to be able to participate because it's higher quality.
All right. Thank you all so much. There were more questions, some more specific to certain companies. So I encourage you to come up and talk to them and ask those questions at when we're done. Up next is the break in the exhibit hall. Please go vote for Best of Show. Those are the new products presented at the event. And thank our speakers again with me.